Hi, everyone. My name is Dr. Jennifer Schramm. I am one of the pediatric cardiac intensivists here at Johns Hopkins Hospital. And today I'm presenting some work that I presented recently from Dr. Alan Everett's lab. And Dr. Everett's lab is focused on biomarkers of pulmonary hypertension and other disease states. So we sought to look at CCN proteins in patients with pulmonary hypertension. So CCN proteins are secreted into the cellular matrix and are important in cell adhesion and chemotaxis, and found implicated in many types of neoplasms, but have also been thought to be correlating with pulmonary hypertension. So we looked at CCN levels. There are 6 of them. We looked at them in 40 adults with pulmonary hypertension and 40 adults without pulmonary hypertension, and found that CCN 1, 2, and 6 correlated the best with just the diagnosis of pulmonary hypertension. CCN 2 correlated best with survival of pulmonary hypertension at 5 years, as well as CCN 6. And then CCN 1 actually correlated well with functional class and 6-minute walk distance, and CCN 6 performed really well against all outcomes, including functional class, 6-minute walk distance, cardiac output, and pulmonary vascular resistance. So what does this all mean? So CCN1 is ubiquitous in the tissues. It's important in angiogenesis, chemotaxis, proliferation, and cell adhesion. And overexpression is actually thought to be protective against PH. CCN2 is also known as CTGF. It's a more common name for it. And it is important in cell adhesion and is a mitoattractant secreted by vascular endothelial cells. And we know that it enhances DNA synthesis and is implicated in systemic sclerosis in some neoplasms. And then finally, CCN6, it has a role in mitochondrial electron transport and mitochondrial respiration, which we know is important in pulmonary hypertension. So all of these together, we hope to create a panel or use them individually to be able to predict disease, to predict an outcome, and then to understand response to therapy. So final work in larger cohorts is ongoing right now. And I would really like to thank the Blalock-Taussig-Thomas Heart Center for supporting all of this work, Dr. Alan Everett's lab and everyone in it for supporting me through this, my PCICU colleagues in the Department of Anesthesia and Critical Care Medicine, and then finally, thank you to Johns Hopkins and the patients that inspire this work.